All right, guys, welcome back to Underground Science. In this video, we'll be talking about sideroblastic anemia. All right, so let's go and get started. So remember to go watch our previous video, which we talked in detail about protoporphyrin synthesis, right? So remember to just go make sure to go and watch that before this, okay? Because now that we have that pathway in mind, right, we can directly talk about what sideroblastic anemia is, right? Since we know how protoporphyrin is synthesized. So remember, right, a quick refresher that protoporphyrin synthesis occurs in the mitochondria of your erythroblasts, right, your developing red blood cells. And remember, inside the mitochondria, iron, right, moves in, right, into your mitochondria, combines with protoporphyrin, right, protoporphyrin 9, and that leads to the production of heme via the help of ferrochelatase. Okay, so we discussed this, right, in our protoporphyrin synthesis video. Okay, so in sideroblastic anemia, then what's the problem? Well, the problem is that you don't produce protoporphyrin. Something is wrong in that pathway where protoporphyrin is not produced. So you don't synthesize protoporphyrin. So the what happens is iron, right, keeps on moving into the mitochondria, right? But there's no protoporphyrin for iron to combine with. So iron keeps on accumulating in the mitochondria of your developing red blood cells, which leads to the formation of ringed sideroblasts, which is basically this iron accumulation, right, in your mitochondria. And these mitochondria, right, these iron accumulated mitochondria surround the nucleus, right, which signifies iron-laden mitochondria, right? So basically, iron-laden mitochondria surround the nucleus of your erythroblasts, which is called ring sideroblasts. All right. So so again, you don't produce protoporphyrin in sideroblastic anemia. So iron, right, keeps on moving into the mitochondria, right, and accumulating, but there's no protoporphyrin for iron to combine with. So you never produce heme. Again, if you don't produce heme, you don't produce adequate levels of hemoglobin, therefore you get microcytic anemia. Okay. So that's how that's the basic pathophysiology of how sideroblastic anemia works. All right. So iron accumulates in the mitochondria of your red blood cell precursors. You get iron-laden mitochondria surrounding your nucleus, which is called ringed sideroblasts. Okay, so let's move on, right? Let's talk about what we see. So what are the causes of sideroblastic anemia? Okay, so causes. So the main cause, right, can be X-linked, so congenital, right? It's a genetic cause where you have a defect in the amino levulinic acid synthase enzyme. Remember this enzyme, with the help of vitamin B6, right, converts succinyl-CoA and glycine into amino levulinic acid, right? So if the enzyme is defective, you can't produce protoporphyrin. If you can't produce protoporphyrin, iron doesn't have any protoporphyrin to combine with to form heme, okay, therefore leading to sideroblastic anemia. Now, other causes include vitamin B6 deficiency, right? If you're deficient in vitamin B6, aminolevulinic acid synthase cannot work, again, leading to no production of protoporphyrin, leading to sideroblastic anemia. Now, a thing to watch out for is patients who are on isoniazid for tuberculosis, for example, right? Remember to keep in mind that isoniazid therapy reduces B6 levels. Okay, so that's another way you can be B6 deficient. And of course, if you're not intaking enough B6, right? Like a nutritional deficiency, right? Through your diet, you're not taking enough vitamin B6. That can be a reason too, right? So alcoholism, for example, there's no nutrients in alcohol, so you can get deficient in B6, which can lead to a defective Alas enzyme, right? Which means the low protoporphyrin synthesis, which can lead to sideroblastic anemia. And then remember lead poisoning. Remember the two enzymes we talked about in the protoporphyrin synthesis video that's affected by lead poisoning. Aminolevulinic acid dehydratase, right, which takes aminolevulinic acid and converts it to porphobilinogen. And ferrochelatase is affected by lead poisoning also. Remember, ferrochelatase takes protoporphyrin 9 and heme and protoporphyrin 9 and iron and combines them to form heme, right? So if ferrochelatase isn't working, you can't form heme. If aminolevulinic acid dehydratase isn't working, then you can't, make protoporphyrin and then therefore can't make heme. Okay, so again, leading to sideroblastic anemia. So those are the main causes to keep in mind for our exams, right? So let's go ahead and move on and see what we have next. So let's look at labs now, since we understand, right, the causes and what sideroblastic anemia is. So for the labs, right, the main labs to focus on, let's start with the CBC, right? So you would see low hemoglobin, right? Why? Well, remember, you don't have protoporphyrin 9. You don't have adequate levels of protoporphyrin 9 levels. And if you don't have protoporphyrin 9, there's no iron, I mean, there's no protoporphyrin 9, right, to combine with iron. Okay, so if you don't have protoporphyrin, right, adequate protoporphyrin levels, iron doesn't have anyone to combine with in order to form heme. If you don't form heme, you don't have adequate hemoglobin levels. Okay, and then of course that means you would see low hemoglobin. Now the RDW in sideroblastic anemia can be elevated, so you can have variation in size of your red blood cells, just like we saw in iron deficiency anemia. All right, but focusing on the iron studies, right? What's interesting is that we call this sideroblastic anemia, right? However, your serum iron is elevated here. Okay, and why does this happen? Right? So it's important to understand why this happens. So first of all, let's understand the normal iron feedback loop. All right. So if you're in an iron overloaded state, okay, if you're in an iron overloaded state, so you have too much iron, right? In an iron overloaded state, would you want to absorb more iron from your intestines to your blood, right? You wouldn't want to do that because you're already overloaded in iron. So in an iron overloaded state, your liver will secrete a lot of hepcidin. And hepcidin, remember, inhibits ferroportin, okay? Remember ferroportin, right? If this is our enterocyte, ferroportin is over here, okay, next to our bloodstream, next to our blood vessel. So what ferroportin does, it takes iron and puts it into the blood. But if you inhibit this, you can't put iron into the blood, and you would want to do that, right, if you're in an iron overloaded state. So that's what hepcidin does. It inhibits ferroportin. Okay, so decreased absorption of iron. And remember, ferroportin is not only in enterocytes, but remember our bone marrow macrophages, right? For example, have ferroportin also. So if ferroportin is inhibited, iron cannot go from inside your macrophages to your red bone marrow. Therefore, you don't produce adequate levels of red blood cells, right? That way you can't produce enough heme. Or this leads to decreased production of red blood cells, I should say. Okay. So that's what happens when that's the function of hepcidin, right? Is what we're trying to get at, is it inhibits ferroportin. All right. So that's what you want to do in an iron overloaded state. So that's a feedback loop, right? High iron, right, leads to secretion of hepcidin, which tries to go and inhibit ferroportin so that you decrease your serum iron levels, right? So you decrease your serum iron levels. Now, the opposite is also occurs, right? If you're deficient in iron, right? So if you're deficient in iron, then what happens is your liver, right, doesn't secrete hepcidin, right? So you have low hepcidin secretion, which means you release ferroportin from inhibition. So ferroportin is no longer inhibited, right? So if this is your enterocyte and you have ferroportin, right, on this side, let's just draw it like that, right? And then your blood vessels here. If you have low hepcidin, ferroportin isn't inhibited anymore, so then iron can get absorbed and go into your bloodstream, right? This is your bloodstream because you're deficient in iron, so you want to increase your iron levels, right? So you want to not inhibit ferroportin, right? Same thing when we talk about your bone marrow macrophages, right? If you're deficient, right, in iron, remember what stage one in iron deficiency? Your storage iron gets depleted first. If you're deficient in iron, you can't properly synthesize red blood cells, right? Because you need iron to make hemoglobin. So then you would want your bone marrow macrophages to release its stored iron so that your developing red blood cells can properly synthesize adequate amounts of hemoglobin, right? So if you have low iron, you would want ferroportin to not be inhibited, okay? Which occurs because in low iron states, your liver produces low hepcidin, right? Which does not inhibit ferroportin releases it from inhibition so that iron can go out and into your red bone marrow so that your red blood cells, developing red blood cells, can use that to form adequate levels of hemoglobin, right? So that's the basic feedback loop of iron and hepcidin. Okay, now if you have increased erythropoiesis, right? So if you have increased erythropoiesis, for example, maybe because, let's write this down, maybe because like you lost blood or you have anemia, right? So your bone marrow is trying to produce more red blood cells, right? If you're trying to produce more red blood cells, you would want ferroportin to work, right? So your bone marrow macrophages can shunt out more iron into your red bone marrow so you can have adequate red blood cell production. So increased demand for erythropoiesis decreases hepcidin, right? Hepcidin levels. Because you don't want hepcidin because you don't want to inhibit ferroportin, okay? Your red blood cells will need iron in order to properly synthesize adequate levels of hemoglobin. Okay, so this brings us to the point of why is serum iron elevated in sideroblastic anemia? Okay, so let's go and see why this happens. Now, remember, in sideroblastic anemia, you're not properly performing erythropoiesis, right? Why? Because you're not synthesizing protoporphyrin, which means you're not able to combine iron with protoporphyrin and make heme. Therefore, if you don't have enough heme, you don't have enough hemoglobin, which means that you're going to have small red blood cells, right? But if you don't have if you don't have protoporphyrin, right, you can't make enough heme and hemoglobin. Therefore, you won't be able to you won't have effective amount of red blood cell production. Okay, so that's ineffective erythropoiesis. So remember what we said is if we have ineffective erythropoiesis, the goal is to have more iron into our blood with the hopes of, right, making ineffective erythropoiesis effective again. Right? So when we have low protoporphyrin levels, we're not synthesizing enough heme. So our body thinks, okay, well, we need more iron, right? So we can synthesize adequate heme, adequate levels of heme and hemoglobin. So what it does is your liver suppresses hepcidin production. This stimulates ferroportin, and you reabsorb more iron from your intestines to your blood, right? So you keep on doing that, and you keep on increasing your serum iron levels. And all that does is it makes more ringed sideroblasts, right? Because iron keeps on accumulating in the mitochondria, right, of your developing red blood cells. So, and then you just keep on increasing your serum iron levels, keep on accumulating and forming ringed sideroblasts, and etc. Right? So it just accumulates in your serum, mitochondria, and your macrophages, right? So you just keep on storing that iron. Okay, so that's the reason why you see elevated serum iron levels as compared to anemia of chronic disease or iron deficiency anemia, okay, where you see low serum iron levels. So hopefully that makes sense, right? Now, the transferrin saturation percentage would be elevated. Why? Because there's high serum iron, so a lot of iron is binding to transferrin. Therefore, you increase your transferrin saturation, right? Which is the amount of how much iron is bound to transferrin. Okay, so that makes sense right? That makes sense also as to why transferrin saturation would be elevated. Now remember, you have a lot of iron, okay? You have a lot of serum iron because you keep on reabsorbing iron due to low hepcidin levels, right? And ineffective erythropoiesis. So all that iron accumulates in your serum and accumulates in your macrophages, right? So that means that increased amount of iron will get stored inside your macrophages. Therefore, your ferritin levels, which is the storage form of iron, is also elevated. Okay. And if your storage form of iron is elevated, you don't want transferrin, right, bringing more iron to your cells, right? Because you already have high stored amount of iron. So why would you want transferrin bringing more iron, right? Your ferritin's already elevated. So it makes sense that your TIBC, which is indirectly measuring transferrin levels, would be low. Okay, so for example, if in anemia of chronic disease, what did we see? We saw low serum iron, right? We saw low transferrin saturation. We saw low TIBC and high ferritin. But in sideroblastic anemia, the problem isn't low iron. Okay, we have a bunch of iron. It's just that iron can't combine with protoporphyrin to make heme. So you have high serum iron, high ferritin, high transferrin saturation. However, you have a low TIBC. Okay, so that's the comparison between anemia of chronic disease and sideroblastic anemia. Okay, so hopefully that concept makes sense, right? So if we talk about so we talked about TIBC, ferritin, serum iron, transferrin saturation. We said RDW can be elevated, and remember your mean corpuscular volume, because you're not making adequate hemoglobin levels, will most likely be low. Okay, so that's why sideroblastic anemia falls under microcytic anemia. Okay, it is said that your mean corpuscular volume in sideroblastic anemia can be normal. Okay, but we still classify this under microcytic anemia. Okay, so MCV less than 80 femtoliters. All right, and if you did a Prussian blue stain, right, you would see ring sideroblasts, right? So we actually did it. If you did a bone marrow biopsy and you did a Prussian blue staining, you would see the ring sideroblasts, okay? So you would see that, because remember, what are ring sideroblasts, right? It's the iron that's accumulated inside of mitochondria, and that mitochondria surrounds your nucleus, which is called, or which are what ring sideroblasts are, okay? So that's a classic lab pattern that we're familiar with now, right? So now the only thing we have to talk about is the treatment, right? Since now we understand what sideroblastic anemia is. So if we talk about the treatment, right? So let's label this slide as the treatment. If we talk about the treatment, right? If you're deficient in B6, right, give B6. Okay. So basically, you're treating the causes. If you're deficient in vitamin B6, we know that the elast enzyme won't work. So give more B6 so that enzyme can work, so you can produce protoporphyrin. So all the iron that's accumulating inside the mitochondria can actually combine with protoporphyrin and form heme, right? So, and then of course, if this is alcohol related, stop drinking alcohol, start eating more nutrition. Okay, that would help also. If this is due to lead poisoning, remember lead poisoning inhibits aminolevulinic acid dehydratase and ferrochelatase, then do chelation, right? So you can decrease the amount of lead in your body. And if this is isoniazid related, right? For example, for treatment of tuberculosis, because that reduces B6 levels, give pyridoxine, right? B6 as a external supplement. Okay. And so yeah, that's basically it. So the treatment is based off of treating the causes, right? So if you have B6 deficiency, right, give B6, whether that's if you're on isoniazid therapy or if you're just not taking enough B6, right? If it's relating to an alcoholic, right? Someone who's drinking way too much alcohol, that doesn't have nutrients, right? So you'd want to stop alcohol, right, and start eating nutrients like B6, right? And then, of course, if it's lead poisoning related, right, you'd want to do chelation so you can reduce the amount of lead in your body, right? That way you can release aminolevulinic acid dehydratase and ferrochelatase from inhibition, therefore producing more protoporphyrin. Therefore, more protoporphyrin can combine with the accumulated iron in the mitochondria and form heme. Okay. And then yeah, that's basically it. So that's the treatment, right? Just focusing on the causes of sideroblastic anemia. Okay. So that's basically it. Thank you guys for watching. Hopefully this video wasn't too confusing. Um, and see you guys in my future videos.